So welcome to everyone. Good morning, and thank you for joining us. My name is Mike Talbot. I am Notre Dame International's Director for Initiatives in Mexico. Today is the third in our Mexico virtual lecture series presented by Notre Dame's Mexico Global Center. For those of you that are joining us for the first time, this series is an opportunity to share information about research and work being done at Notre Dame by Notre Dame faculty, either on, in, or with partners in Mexico. Our main audience is really the Notre Dame community in Mexico, which is quite large, consisting of students, parents, alumni, all very interested to know more about the frankly deep connections between the university we love and this country that we also love. So here today to join us and share a little bit about their work in Mexico are Doctors Nicole Achi and John Grieco. Before we begin, I just have a few requests. First, please keep your microphones muted. This will just eliminate background noise from interrupting the presentation. And as someone who lives next to a busy road, I can tell you that can be a problem from time to time, so I'll be practicing that as well. Second, we do invite you to turn on your video. It was probably off when you came in, but it's great to see everyone's faces, so if you feel comfortable doing so, please turn on your video. Third, today we're going to try a new format for questions and answers. We're going to use the raise hand feature. For those of you not familiar with this, if you press the participants button at the bottom of your screen, another window will pop up, and on the bottom of that window there's an option to raise your hand. So simply raise your hand, and at the end of the presentation we'll go through and unmute your microphone so that you can ask your question yourself. If for whatever reason though that's not working or you're not comfortable doing so, you certainly can still submit your question via the chat feature. So today we're very pleased to have with us Doctors Nicole Achi and John Grieco. They are both research professors in Notre Dame's Department of Biological Sciences, as well as the Eck Institute for Global Health. Together they oversee the Achi-Grieco Lab, which focuses on the biology, ecology, and transmission dynamics of vector-borne illnesses such as malaria, dengue, chikungunya, I always mispronounce that, and other diseases as well. In addition to their extensive work in Mexico, which we'll be learning about today, they also conduct research with partners in Belize, Indonesia, Nepal, Peru, South Korea, Suriname, Tanzania, Thailand, so basically everywhere. Their research has appeared in the American Journal of Tropical Medicine and Hygiene, the Journal of Vector Ecology, the Journal of the American Mosquito Control Association, and several others. I just have to say, I was fortunate enough to join them when they visited partners in Monterey and Chiapas this past January. It was really great to see the extent of their commitment to collaboration and partners here in Mexico. So without any more ado, I'm going to hand it over to Doctors Achi and Grieco, and thank you both very much for joining us today.
Great. Thank you, Michael. Well, I will start off the presentation, and John will have the second half. And I'll be turning off my video in a moment because, of course, I'm sharing my screen. But I just wanted everybody to be able to see my face as we go through the presentation. But first, just let me thank you, Mike, for extending the invitation. We're excited to participate and raise some awareness, as you said, of how Notre Dame has been or is envisioned to be continuing to collaborate with the Mexican partners. Can you hear me okay? That's first. Okay, great. So let me just hide this. So yeah, so we're both in the Department of Biological Sciences. As Mike just explained, we are medical entomologists, which means that we focus our research on insects that cause human disease. Oh, excuse me, of course. I will have to, can you still see my screen? Michael? Can you still
Yeah,
hear me?
we can
Yes.
see.
Oh, okay.
It's not
Okay, okay.
yeah.
Yeah,
Yes, we
I'm
can
gonna
see you.
have, some reason my slideshow stopped, so I'm just gonna have to do it this way. Oh, well, it's not advancing, of course, as we just tried that, and now it's not advancing. Let me see here. There we go. Okay. So what was today's ask by Michael? It was basically that Notre Dame International, and specifically the Mexico Global Center, Mike expressed this statement, which was, now more than ever, the students, parents, and alumni that make up the Notre Dame community here in Mexico are curious to learn about the connections between the country and our university. And as I said, we're delighted to be participating in this presentation. And we hope that we have outlined at a very high level how we have made connections or are hoping to continue in expanding those connections. So as we just mentioned, we are medical entomologists by training, but what is the focus of our research? Our ultimate goal really is to provide safe and efficacious vector control products. And I'll explain what a vector is and what we mean by vector-borne diseases in the next couple slides. But the purpose really of the research is to incentivize research and development innovation by corporate partners for improved disease prevention, making better products or optimizing the products that we have available to us to prevent human diseases that are caused by insects. And ultimately collaborating with stakeholders to enhance the capacity building and shaping health policy. John and I are passionate about having truly active partnerships in all of the work that we do and research that we do around the world. Mexico, of course, is part of that. And the underlying foundation is to improve the quality of life of local communities. So the approach that we have taken for making these connections and renewing friendships and expanding our relationships in Mexico is the same approach that we take for all of our work. And that is that remembering it's always bigger than you, it's bigger than us. So we are always identifying partners in country that share the same vision for the focus of our research and the ultimate goal that I just explained in the previous slide. And we always look for ways of understanding better our partner needs and surrounding politics and governance structures within the countries that we conduct research. And that really helps to manage expectations. You want to forge relationships. This is for the long term. You want to build that respect. And then engage with collaborators in different ways. You do have to be innovative, especially look at what happens right now with the COVID-19 pandemic. So although our research is scientifically based, we still look for ways to be innovative in our collaborations, which includes training, student exchanges, and John will talk about that. We are looking to promote and build relationships that go beyond just the 1 or 2 year projects that we are able to get funding for. And then promote equity towards capacity strengthening. So even though Notre Dame is a prominent university, we're very proud to be part of the university faculty, but we always can learn from one another at a global scale. So the intention is to have this cross-learning and cross-building. So the presentation, we've broken it down to 4 parts. I'm gonna provide a little bit of context and background to the work that we're doing in Mexico, and I'll explain briefly on some current research, and then John will follow with programs and development, and lastly, discussing a little bit about our capacity strengthening and sustainability partnerships in country. So first, context and background. So what are vectors? What are vector-borne diseases? And in simple terms, a vector is an organism that does not cause the disease itself, but which spreads infection by conveying pathogens from one host to another. In our circumstance, our training, as I mentioned, is in medical entomology. So our vectors are insects such as bugs and mosquitoes and ticks, and there's others that are relevant globally for many current and emerging human diseases. So for example, if we look at mosquitoes, some of you may have already seen this slide. It's from the Gates Notes. Bill and Melinda Gates Foundation puts out these notes. And maybe about 4 years ago, they had this information that the world's deadliest animal actually is mosquitoes. It is that group of insects is responsible for causing about 800 to 900,000 deaths in humans per year. And arguably the more important of the mosquitoes are the Anopheles genus and Aedes aegypti species of mosquito, which causes malaria from the Anopheles and arboviruses such as dengue and chikungunya and Zika from Aedes aegypti. They vector different diseases, different pathogens. If we look at the impact of malaria deaths per year, just to put it in some frame of reference, here in the US, if we look at per year deaths caused by malaria, it would be equivalent to saying all of the population of Portland, Oregon, all of the population of Honolulu, Las Vegas, each one of those, each year is what it equates to. And if we want to relate it to Mexico and maybe Monterrey, Monterrey has at least reported about 1 million individuals population density. So that would be about half of the population of Monterrey every year dying from malaria alone. And for dengue, which is an arbovirus transmitted by Aedes aegypti mosquito, the impact of dengue per year is about 100 crashes of a 747-700 jumbo jet with 520 passengers each time. So about 500,000, but there's many more at risk, not just death, but at risk of having severe sickness. So very severe, very important vector-borne diseases. Another group of insects, ticks, are responsible for another large variation of pathogen transmission, including bacteria such as rickettsiosis, spotted fever group, rickettsiosis being probably arguably the most important worldwide, because they can, this particular bacterial infection can cause up to 5 to 10% case fatality rate if not treated. And it's important to state that for rickettsiosis, it's a simple treatment of antibacteria medication, so antibiotics. But the challenge is having people come in, have access to healthcare, and the local physicians recognizing that it might be a rickettsial disease, say as compared to dengue or other types of pathogens, in treating that properly. But for tick-borne rickettsiosis here in the US, we have only been on the increase in terms of the number of cases being detected and reported. And so it's a very important vector-borne disease, and that includes in Mexico as well. So the challenge that we have as medical entomologists, and the research why we're doing this research, is that not all vectors are the same. Not all anopheles are the same. So the malaria changes depending on where you are in the world, and severity and the type of vector that's transmitting it, which anopheles. Same with Aedes aegypti. All Aedes aegypti transmit dengue, but the transmission dynamics can vary. So not one strategy or product can be as effective across the world as in another location. And so we have complex settings too, maybe refugees or internally displaced persons that can create a challenging environment for trying to control for vector-borne diseases. And so we have to adapt and work with our partners in country to, as we mentioned, determine their needs and their challenges, and how can we work together to find a resolution of new products and strategies for these vector-borne disease prevention goals. And here's just one example. This is from Brazil, but there in Mexico, there's also examples of where you have very complex settings in terms of urban environments. You have limited resources where perhaps governments and academic researchers may not have all of the means that they need or require for effectively combating such vector-borne diseases, especially, say, dengue. So the urgency and relevance of vector-borne diseases, there's just some examples that we put into our presentation for various types of vector-borne diseases around the world. Zika virus, I think most of the people perhaps that are joining us today have heard of Zika. That was a major outbreak maybe 4 years ago. It had a lot of sequelae, meaning microcephaly in children, long-term health burdens in these countries, not just death, but long-term care. And that can occur also with dengue and malaria, those that survive. But we have malaria outbreaks occurring in Africa. Yellow fever is another Aedes aegypti virus that's transmitted by Aedes aegypti. Severe outcomes in that group of diseases. And then in Mexico, of course, dengue is one of the more prominent mosquito-borne and vector-borne diseases of importance. And right now, there's been an increase in the number of dengue cases being reported as compared to the past year, 62% higher in some locations in Mexico than previously. Oh, I don't know why that's occurring, excuse me. So the WHO has put out a statement saying that vector-borne diseases has accounted for more than 17% of all infectious diseases. That's all of the infectious diseases is caused by vectors. Another 2/3 are zoonotic. So we put this statement out there because it's important to stress that these are globally important diseases, and it accounts for a high percentage of all infectious diseases around the world. Let me see, I don't know why that's not. Okay. So that's just a little bit of context and background. So I'm just going to give a couple slides on the current research that we're conducting in Mexico. And here's where we want to acknowledge our in-country partners to date. And these are active partners. They represent the Ministry of Health, clinicians, academics. So there's a wide variety of expertise in a holistic approach. The first individual I would like to recognize, who might be joining us actually on the seminar Zoom session today, is Dr. Ildefonso Fernandez Salas. He is based at the University of Monterrey, Autónoma de Nuevo León in Monterrey, but he has worked within the CRISP, the Public Health Research Institute in Tapachula in Chiapas as well. And really him and his students and his team have been gracious hosts to introduce us to some of the other partners that we've been collaborating with. And just to note that Ildefonso actually is a long-term friend. So we've known Ildefonso for about 20 years, if not longer. We graduated from, John and myself and Ildefonso graduated from the same university with our doctorate degrees. So it's a long-term relationship that we have, and we're really pleased to be able to initiate some of our research programs with him as well and his team. The other individual who might be with us is Julio Adonis Lozano, who is the director of the Public Health Research Institute in Tapachula in Chiapas. And him and his faculty and his students have been gracious as well. And we've been working with them to beginning to develop programs and thinking about planning for future capacity strengthening within the region. And they both have been able to introduce us to Dr. Jesus Sepulveda Delgado, who is the coordinator of research at the regional hospital in Tapachula. Dr. Jesus has been very interested in learning how Notre Dame proper, and of course through maybe myself and John specifically, might be able to forge some interesting and adaptive creative ways of getting training for his residents in science, scientific research and research method. So there's been very interesting conversations with these active partners, and we're thrilled to be able to have these relationships. So the first project that I'm just going to mention, and then I'll turn it over to John, is our what's entitled a biologically informed framework for vector surveillance in northern Mexico along migrant routes. Very long title, but the focus is to understand better what types of vector-borne diseases may be circulating within Mexico in specific targeted areas where we might see a lot of people moving from other countries into Mexico or from Mexico into other countries. And this is really meant to serve as some surveillance information to help public health officers, clinicians be better aware of the types of diseases that may be presenting in cases when people come in to seek healthcare. So this program is supported by the U.S. Department of Defense. It's the Global Emerging Infectious Surveillance Program. And we partner, John and myself at the University of Notre Dame, we partner with the Navy Medical Research Center, the U.S. Navy, along with Dr. Ildefonso and Dr. Julio, so CRISP and University Autónoma de Nuevo León. We are all working together to establish this program. It is funded. It has been put on delay just because of the coronavirus at the moment, but it is an active program. And this is focused, right now we're focusing on tick-borne rickettsiosis. As I mentioned, those are very important diseases of humans. Mexico is endemic for one of the more important tick-borne rickettsiosis, which is Rocky Mountain Spotted Fever. We also have that here in the US. And previous studies have indicated either the tick has had the pathogen of RMSF, the bacteria, or there's been cases that have been diagnosed with RMSF, Rocky Mountain Spotted Fever. So we know that it's in Mexico. Historically, it's been reported. What this GEIS program will be doing is gathering information in targeted areas in Chiapas and also in Nuevo León, as I mentioned, where you might have border crossings. So that way we have a better understanding as to the prevalence and how best to identify which ticks are responsible for transmitting, so we know how best to control ticks from biting individuals in that area and spreading the disease. This is just an example of where we see a lot of movement of people and populations at the southern border of Mexico with Guatemala. And again, we have another active border between the US and northern Mexico and Nuevo León. And this is where we intend to do some sampling in Mexico for ticks. So how do we do that? We'll be partnering with veterinarians to help collect ticks off of horses and cattle. We'll also do tick drags, which is just a cloth sheet that you just drag across the vegetation. And you passively look to see what ticks might be in particular areas around farms or even around semi-urban environments. And we'll have the samples being processed in partnership with the Navy Medical Research Center, also through training at the CRISP in University Autónoma Nuevo León in Monterrey. So we can identify what the tick species is, whether or not they're infected. And that's very important. So we understand the risk of infection, but also how do we help to prevent biting from a particular species of tick that may be that people may be exposed to. And so this program will last for 2 years, and we're hoping that from there we will be able to build onto more research that's related. So from there, I'm going to turn it over to John, and he's going to talk about programs that we currently are developing, that we're writing, and then lead end the conversation with capacity strength and sustainability. So John, I'm going to just go ahead and pass it on to you. Just let me know when you'd like me to advance.
Great, thanks Nicole. And thanks Michael for the invitation to speak today. Also thanks to all of our colleagues in Mexico, as well as all of the participants on the seminar today. I think this is a really exciting venture that Michael has set up for kind of introducing everybody to what work Notre Dame is doing in Mexico. And I think this can only strengthen the efforts as we move forward with our collaborators. But thank you again to everybody. So as Nicole mentioned, there's a number of things that are kind of envisioned or in the works. We're talking with our collaborators in Mexico, Ildefonso, Julio, to kind of move to the next step. I mean, obviously the GEIS grant to do the screening of the ticks is kind of the first step on building some of these relationships, getting networks set up. But while we were in Mexico recently, on that trip that Michael talked about, there was a number of discussions that we had about what are some areas that we might move into? What are some of the things, listening to our Mexican colleagues to find out where the needs are, and what would be the interest level for moving forward and having Notre Dame as an active partner in getting some of these initiatives done. So Nicole, if you can advance. So I think one of the areas, yeah, this is perfect. So one of the areas is a training program using a digital education platform. You know, we spoke with Dr. Ildefonso about, you know, he's set up some of these things at his university and has already dabbled in getting some of this digital education platform set up. And so we thought, you know, that could be an area that's fertile for us to move forward together with our Mexican colleagues to develop a more of a digital platform for medical entomology training. This could be offered to students. This could be offered to technicians that may not have access to, being able to come up to formal universities to get training, but be able to provide that kind of underlying foundational science throughout Mexico for people who wanted to log on and who could have access to training materials to build on that foundation, to build that next generation of in-country medical entomologists, to spur them to become more interested in going to universities to do this, but also to do research at some level, whether that be a technician level or carry on to get a higher level degree. So we put in a small seed grant program through the Notre Dame International. And we'd like to again thank Michael and as well as all of Notre Dame International. They just awarded this like a week ago. So we have about a sum of money to be able to move forward in developing this. But we've also been in contact with our own center at Notre Dame, the Eck Institute of Global Health, and they are also interested in putting together additional funds to make sure that this initiative is moved forward. So we look at this as a platform for Mexico, but then it could be expanded to more of a regional approach and get more regional partners involved, such as Belize, Guatemala, El Salvador, as we expand out. So Nicole, can you advance? So this is kind of in a quick diagrammatic form, what we envision for the first year of this, because the funding right now is for 1 year. But we looked at, as we gain traction with this, we can look for other methods of funding as we move forward, other grant possibilities, and hopefully expand into subsequent years. But in year one, the training format is really to digitize some of the medical entomology courses that we have, translate them into Spanish, and then convert them into a partial hybrid approach where we can house them maybe at the University Autónoma de Nuevo León with Ildefonso and have them out as a digital platform where maybe some face-to-face kind of training can go on with more of like a laboratory-based training, things like insecticide resistance training, other things that we do as medical entomologists that are core to our ability to protect those communities from vector-borne diseases, but may have a little bit of a challenge with getting out to rural communities or doing not in a face-to-face fashion. And actually, some of this COVID situation is actually really preparing us for moving this initiative forward. Notre Dame has not been, had a lot of the digital education platform going. They've got experience in it, but they really haven't invested a huge amount into that platform. But now with COVID, a lot of the technical aspects are really getting worked out. Then we're also going to be relying on our digital education group at Notre Dame to improve on how do you develop the metrics around evaluation of these training programs. How do you evaluate the students' progress? How do you determine whether they're really taking up the material and being able to apply it? So Notre Dame does have a wealth of experience in that, working with groups like IBM Watson to develop a badging system, a micro-credential system, where we can evaluate whether the student is truly gathering the core tenets of those digital education platforms and being able to apply them back out. Not just rote memorization or, a student watches a video and then 3 days later they forget the material. So we're really going to concentrate on making sure whatever is put out by these programs will then be able to be uptake by the individual and then be able to put back out. And again, this will be geared towards a broad audience in Mexico. Maybe some college level students, but in addition to that, more the technical people, the people on the ground that are doing this in Tapachula or remote communities in Mexico will have access to this. And this is not this will not be a paid program. This is a free program that the students will be able to have access to and be able to get some type of credentials or badging or certificate at the end to make sure that they get credit for these types of courses as it's moved forward. So that's what we envision in kind of year one. And we're already in discussions with other groups, as well as looking at other granting options to carry this beyond the 1 year kind of mechanism that we currently have in place. But this is already, we already have the funds for this, and so this will be moving forward. Right now, again, because of the COVID and some of the uncertainty around, the economics of this, we have been asked to postpone until, at the earliest January of this next year. So it gives us time to discuss with Ildefonso and Julio and other collaborators within Mexico to find out what are those course materials we really want to provide, and how do we structure this moving forward to make sure that this is a success. Okay, Nicole. The other really exciting initiative that I think is kind of on the edge of being funded, and I think we're really looking at seeing how we can move this forward in Mexico as well, is we're calling it the Remote Emerging Disease Intelligence Network, or ReadyNet. And this plays off of a little bit of what Nicole was talking about with the rickettsial study going on with the DoD. Again, this is another DoD funded, or would be a DoD supported initiative, but it's also working with groups like NASA, groups like the Smithsonian, the Navy, and Notre Dame as being the lead partners in all of this to develop a kind of a screening system that's not just focused on Mexico, but more globally. And one of our key partners we're hoping in the future will be Mexico to have as part of the outreach laboratories for evaluation of where some of these pathogens are being transmitted. And when we originally envisioned this program, it was focused around vector-borne diseases, but then COVID-19 hit, and we broadened that to where we're going to be looking at things, not only the vectors in various regions, but also what's called eDNA. And eDNA is environmental DNA. So if you envision, say, a watering hole, someplace where large numbers of wild animals come to this common source to drink, they're going to be defecating, they're going to be urinating in that area. They're also going to be drinking the water. So you're going to have saliva and antigens that are going to be introduced into that water. Well, a lot of the technologies now are such that we can actually go and test the water source. We can test the soil samples, look for this eDNA, and find out if there are viruses, if there are pathogens, not just one particular pathogen. We can look for dengue, we can look for things like COVID. And by taking these samples and analyzing them, we can determine if there is a threshold within these animal commonplaces that may be spilling over into the human population. So we're trying to get a much larger group at Notre Dame involved that are looking at things like antimicrobial resistance, and as well as things around the environmental impact of water sanitation. So making this a more of a holistic approach and seeing what kind of pathogens are circulating. Then we'll use our partners. We'll have modelers involved who will look at thresholds. When will these pathogens reach a threshold in the environment and spill over into the human population? Thus, we could identify maybe potential for things like COVID-19 before they actually become a problem. So again, focusing much broader beyond this initiative for vector-borne diseases, but into other pathogens and other diseases that may impact on humans that we may not even know about at this point. But one of the nice things about this is it's a capacity building within this program. We're developing kits, and there's as you can see from this diagram, there's a device called a MinION. It's a small RT-PCR unit that can be deployed into settings so that in-country partners can actually test real time. The samples can be collected, put on this MinION device, and it's a very inexpensive kit that we're developing, probably around $2,000 per kit, that we would provide to our in-country partners, do all of the training. They could then do the sampling and testing on site, and then upload that into a virtual cloud. And we're trying to develop a dashboard where we can actually get that data out and show risks of various diseases occurring real time. And so this is what's getting set up. So Notre Dame, the Army, and the Navy will be the reach-back labs. And what does that mean? That means we will do the validation and the verification but we will provide these kits to our partners in a variety of locations. Right now, we're looking at Mexico and Central America. We're looking at parts of Africa as well as Asia to beta test this. That way, when the samples come in, they can test them. They'll send part of that sample back to Notre Dame. We will validate, and then eventually, the in-country partners will be on their own. They can start doing all of this real time, uploading the data, and then we will serve as a conduit and basically a verification tool for them if they need it. In addition to that, NASA is developing a mobile identification tool. We will provide cameras, digital cameras, as well as AI software that a student in Mexico or in Kenya can can videotape or actually take a snapshot of a tick or a mosquito. That will be fed into an AI identification process, and we'll be able to identify real time what those mosquitoes are, what those ticks are on site. So there's a lot of moving parts to this initiative, but in every part of it, we will be needing to work with our overseas labs, Mexico, Kenya, Korea, to be able to utilize this platform and create and generate the data back and forth. So I think this is a really kind of exciting initiative that we're just in the beginning stages of, and we're hoping to be able to have some type of training and discussions with our colleagues to figure out what is the best path forward to make sure that this is a reality. Okay, next slide. So then, we really are interested in making sure the capacity strengthening and sustainability of all of these initiatives that we're talking about. We see this over and over that, universities will come to a location, they'll take all those samples, they go back, and nothing is left in country. And that's just not how Nicole and I operate. We want to make sure that the in-country partners can do and stand on their own with the same technology that we're doing back home, because that's the only way for these initiatives to move forward. If it's constantly relying on an in-country Notre Dame institution to get the data, to analyze, that's not sustainable. Our in-country partner has to be doing this side by side with us, and we can help with training and we can help stand up some of these things from a financial standpoint as well. But that eventually all of these activities have to be done in country. Okay, Nicole? So we like to use the training model that we use in Belize. We have Nicole and I have an active research program in Belize as well, where we've set up the Belize Vector and Ecology Center. Part of that is training, it's developing research programs, it's working with universities, but eventually all of this will be turned over to the in-country partners. We don't see this as being Notre Dame coming down and just basically taking over the research. It's making sure that the capacity is there so that we can relinquish all of these efforts in the near future so that all of them can be done by our in-country partners. And that's kind of how we see this with all of our international sites, is that we're building that capacity, we're making sure the training opportunities are there, we're making sure that the infrastructure is set up, and then we can back off and let the program run on its own. Okay. Nicole, okay. So part of this, and again, this is something that we've had a lot of interest from our international or our domestic partners in coming to Mexico and actually getting this done. We have an initiative called the IMPACTS Initiative, which is Integrated Mayo Program for Arbovirus Community Health Training and Surveillance. Again, big mouthful, but it's a catchy phrase. So our Mayo, when we talk about Mayo, is our Mayo clinicians at the Mayo Institute in Rochester, Minnesota. World-renowned, and these are also the two people we worked with at Mayo are both Notre Dame graduates. So they see the impact of what Notre Dame can do in a lot of these locations. And here we have healthcare provider training. Thus the reason why we're trying to engage with Jesus in Tapachula. We're also looking at vector surveillance and human surveillance. So again, our CRISP partners, our partners with Ildefonso, to make sure that they have the ability to train on the latest diagnostics coming through Mayo for human samples. We can help do some of that for the vector samples on the Notre Dame side. Also, community health education, working with the community healthcare workers, and then vector surveillance and risk mapping. Some of the things that we're doing with Ildefonso and CRISP on the tick side. So again, we've already contacted our Mayo colleagues. They're just trying to figure out when is the best time to come down, have a much more robust dialogue about how do we engage in doing some of the same things that we're doing in Belize, but doing that in Mexico. And again, this partially is funded already. This is most of the funds that come for the Impacts Initiative are from donors from Notre Dame, donors that want to see these types of initiatives carried out in their host country, but also ones that want to do good around the world. So these are donor funds, and they're already available to see how can we make this a possibility in Mexico, and using the Belize model as to stand it up. Okay. And part of the Notre Dame International has already provided some seed funding from a previous grant, and that's how we were able to have our initial meetings at CRISP. Nicole and I and some of our Belize colleagues came to CRISP, I guess it was about 3 years ago, and engaged in how to make some of this a reality. And that has then led to some of the furthering NDI money, but also some of these other grants, like through GEIS and some of these other initiatives. So again, we're thankful to NDI for providing some of this seed money. But it also has provided money for some of our Mexican colleagues, Kayla and Nacho, that work at CRISP, to come up to Notre Dame and do some training with us. And again, I like how Nicole put it, is this is not a one-way street. They are coming up to do some specific training about things that they may not have access to in Mexico, but our students, our researchers also benefited from having Kayla and Nacho, because there are things that they have experience with that we at Notre Dame just don't have. So it's a constant dialogue back and forth between our colleagues as well as theirs, to make sure that we're getting this cross-fertilization of ideas, of trainings, and it's been really productive for us at Notre Dame, but also I hope for our Mexican colleagues. And then from the training standpoint, we've also sent a number of students, most of them from the Global Health program at Notre Dame. We've had people such as John Nida, who is now a physician in Michigan, who did his work on genetically modified mosquitoes and kind of the perception of genetically modified mosquitoes and their use in Mexico. We've had other students like Darlene Kim, who is now also a dentist. She came down as part of her Global Health program, but she assessed different things around onchocerciasis in Chiapas early on in the program. And then we had people like Anna, who was doing head lice studies with Ildefonso down at CRISP. So again, a very diverse group of students, but each one of them came back with a glowing vision of what this program could have for Notre Dame, and hopefully kind of this cross exchange of students with Mexico, so that our students could get better idea of what's going on in Mexico, but also for Mexican students to come up and train at Notre Dame. And we see that this can actually be advanced here in the near future, because we're hoping that these will set the tone for things like training grants. There's a number of global health infectious disease training grants that are offered through NIH, but they require a kind of a long-term vision, a history of training that's been going on. And I think all of these activities that we've got going on with Mexico build that foundation, so that when we go for an NIH grant, people say they have that continuity there. We want to invest in this. And so I think in the near term, we can actually begin looking at some training grants that will allow Mexican students to come to Notre Dame on a full scholarship, but also for some of our students to come down and get additional experiences in Mexico. Okay. So in closing, there's a lot of going on in vector-borne diseases, and it represents a major health burden in Mexico as well as in many other countries of the world. Many ND Mexico research and capacity strengthening programs are in place. And there are also a lot of initiatives that are being planned and talked about with our collaborators, because we've got wonderful collaborators in Mexico, and a lot of good ideas that they have about what they need or what Notre Dame could help augment as we move forward. In successful implementation of these and the health improvement strategies relies on this good partnership. But it's not just a partnership with our academic colleagues, but also with the ministries of health, with industry partners. So it requires a broad approach. And I think that's what we're building, and we continue to build in Mexico, is this kind of cross-disciplinary approach. And with some of the trips that NDI is kind of helping with, helping us establish down in Mexico, I think it's just a really valuable thing to have the support of NDI, but also groups like our Global Health Program and Bernard Nolan, who are really trying to develop more of a touch point in Mexico and develop these programs, because it is such a wealth of talent in Mexico, and everybody sees that. And we'd be very remiss in not engaging in our partners to the south. We also just want to make sure that people understand that, these diseases are not going away. As you can see with COVID, that's just one example of how these things can impact on our health at a global scale. And we all need to be an interconnected partnership. So as you heard from Nicole, maybe some of these diseases are new to you. But even as we talk about that, there are things that are on the horizon, things like Powassan virus, things that are transmitted by ticks here in the United States that we only recently found out about, maybe 2 or 3 years ago. Things like Myaro, other viruses that are circulating that are out there, but we really don't know much about, and we're only now finding these things out. And thus that DoD initiative that we're talking about will really help us get kind of a jump on these diseases to find out what's going on, what may be impacting our communities as we become more and more globalized as a population. Okay, Nicole? So I think with that, I will stop, and I know there's probably a lot of questions, and I hope we can answer them. But again, I want to call out all of our Mexican collaborators who are, I think, on the call today. Ildefonso, Julio, Mauricio, Dr. Rosa, and there's a whole group of great partners that we have, and I hope that we can continue to expand these relationships into the near future. So with that, I will stop, and I will open it up for questions to myself or Dr. Nicole. Thank you.
Thank you very much, John and Nicole. It was a great presentation. Great to see not just one, but multiple really strong projects and deep connection and engagement with Mexico. Just a reminder to everyone, if you'd like to ask a question, feel free to raise your hand using the raise hand feature, which is at the bottom of the participants window. If you're unable to do that, you can certainly submit it via chat. I'll go ahead and get us started with the first question. I think it's a requirement now that every webinar has a COVID-related question. So I'm going to ask that, just get it out of the way. I'm thinking of the restrictions, the shutdown, the social distancing that's happening. And I'm wondering how you think that'll impact vector control. So I'm thinking about issues with household spraying. Are we in a situation now where our very important and understandable fight against COVID is creating challenges for controlling vectors and perhaps seeding some future challenges in some future challenges?
Yeah.
Yes,
Nicole, you want me to just take a quick shot at this? Yeah, so we actually do have a program with a situation where we're dealing with malaria control in Kenya. And one of the challenges that we're seeing is that there's multiple challenges. One's from a social science standpoint, where people are becoming more and more fearful that if they have malaria, are they going to be more likely to catch COVID? So there's a big press on the government to provide more tools and more, things for them to control malaria, given that they think that they may be at higher risk if they get COVID. And so just from an educational standpoint, getting people aware of what those challenges are and what the truth is out there in terms of transmission and what it is, the risk to their health, but also availability of commodities. As you have these restrictions in travel, typically you'll get millions of bed nets transported to Africa on a monthly basis. Now ports are closed, planes are not delivering. So these commodities are no longer available. Many people are no longer getting those vector control tools that will protect them from things like dengue or malaria. And then another situation are the diagnostics. So if you think about a lot of the things that go into the diagnostics for COVID, those are the same things that are going into the diagnostics for malaria, for dengue, you know, these test kits. And so as the shift goes towards mass producing of kits for diagnosis of COVID, many things begin to wane on the malaria front or on the dengue front. And so you get shortages. So this is making the whole vector control arena completely challenged, and it's much more complex because all resources are now being mobilized for COVID, and you know, rightly so. But there's at the detriment, or at the, you know, as you develop these vacuums around some of these other diseases. So it's a challenge. So Nicole, would you know?
Well, I agree. I agree with those points. I mean, we know, as you mentioned from other studies and other projects, that there are challenges in getting the supplies, perhaps, and even, you know, access to care. But I will say this, that through these difficult times, I do believe that there's also some really interesting innovation that is being discussed. In other words, we have perhaps new technology about contact tracing that can then be further applied to informing or raising awareness or cueing people about, you know, other types of diseases, or saying it's time to come in for your check, you know, for say, you know, rickettsiosis, this is the rash that's occurring, you know, and they can do this virtual conversation perhaps, you know, with their healthcare providers. And that's not in every country, in every setting, but I just think it's important to kind of balance out that there could be some really innovative technology that could be coming into vector-borne disease prevention and control from the coronavirus pandemic conversation. And I just bring up the idea of this virtual, you know, healthcare access. That could be quite interesting. Many people around the world, even if they're in remote settings with limited resources, still have a cell phone. That's the interesting part. And so it could become maybe an interesting conversation about vector-borne disease prevention and control, and access to healthcare through a virtual platform that we didn't have before. So I just want to make sure that that's mentioned.
Thank you, Nicole, for keeping us optimistic.
I mean, it's definitely, yeah, it's a different time, but I do believe that there will be some innovations that will come out that could be very beneficial for vector-borne disease prevention and control as well.
Wonderful, thank you. I see that Ildefonso has unmuted his microphone. Did you have something you wanted to add?
Oh, yes, yes, to supplement this very nice, by the way, congratulations, Nicole and John, for this very nice talk. To supplement with some comments, see, I feel very pessimistic about the future because, you know, for instance, in 2002, the first SARS virus arrived, 2002. Then in 2012 was the MERS, which is a second type of SARS. And now 2020, SARS-CoV-2. 3 pathogens plus. In 2009, we got the H1N1 virus, another pandemic. In our field, vector-borne diseases, we got 20 years ago, West Nile virus. I remember that. And then dengue was the largest number of cases were reported last year in Latin America. Now we got 2014 Zika, and no, chikungunya, and 2015 Zika. So these are the pathogens emerged in the last 20 years. So the future is really bad. I mean, after being confined in my house for 2 months, I think, I mean, I cannot believe, I mean, this cannot happen again. It's not possible. I mean, it costed so many lives and the economies for many countries. I mean, it's hard to believe that this happened. But the future looks not very optimistic, because we have yellow fever, outbreaks in Africa and South America. We still some Ebola active, Lassa fever, other. I mean, if you check the PubMed reports, all these pathogens are around. So what will be the next pathogens emerging in the next 5 years, for instance? So I think the role of Notre Dame in this initiative of starting to prepare human resources is very good, it's very welcome, and really deserves more funding. I mean, it's good that we share resources and to start this digital network to educate new people, new human resources. I really like the initiative of John and Nicole because they are making a big effort to push ours in Mexico, our friends in Korea, the good friends in Sri Lanka, and our guys in India and other countries. It's very important that vector-borne diseases being part of the infectious diseases have more support, definitely, and that's nice. I mean, I really like the initiative of the Mayo people, and I like all this work of you, Mike, in Mexico, which is really good. So, I mean, we should be aware, and that's nice if Young and Nicole continue this effort. We will be very happy to support all of you in our humble efforts here in Mexico and in Monterrey and in Tapachula. Thank you.
Yeah, thank you, Ildefonso.
Thank you.
Those very nice words, and definitely, I mean, the support that yourself and all of your colleagues, Julio and Dr. Rossi and Mauricio, everybody has just been a wonderful working partner, and Nicole and I can definitely see working with you guys, you know, into the long future. So we really enjoy working with you guys.
I'll just add my appreciation. I think that was a great intervention, and you know, it's also great to see so much collaboration input from Ildefonso and all the other partners. I'm going to take advantage of this opportunity to ask another question. As a layperson, a non-scientist, you know, I've found the work that you've done or you've discussed, you mentioned briefly in your presentation, with genetically modified mosquitoes in Chiapas and elsewhere, really interesting. Do you think you could just share with us, I think many people on the line might find that interesting as well, a little bit about what that is and how it works or is supposed to work, in theory at least?
Nicole?
Oh, sure. So just for one quick clarification, at least, is that the work that, the studies that John mentioned in terms of genetically modified mosquitoes in Mexico and Chiapas was strictly a questionnaire, a survey that was conducted to gain insight on some initial perspectives on the release of genetically modified mosquitoes. So I just want to make sure it's very clear. So we did not release any genetically modified mosquitoes, but certainly that is one strategy that is being explored and investigated around the world for both dengue prevention and malaria prevention. Now, if you remember in my initial slides, those are two different mosquito groups, one that causes malaria and one that causes dengue. But specifically in Mexico, really the focus of a potential strategy of a genetically modified Aedes aegypti for dengue prevention. This is all very much under conversation, and Ildefonso and others can add to what I'll be saying because they'll know on the status in their regions. But the intention is that if you introduce a naturally occurring bacteria that is found in many insects, it's called Wolbachia. And that's one strategy that can be used to introduce bacteria into Aedes aegypti from another mosquito. And it can help prevent dengue virus from actually developing in the mosquito, and therefore the Aedes aegypti that has this bacteria cannot transmit. So that's not necessarily for some people considered genetically modified, but it is in some way a modified mosquito that's being used to explore using those bacteria-infected mosquitoes released into the environment, and it may prevent transmission of dengue. Now, for the true traditional genetically modification of mosquitoes, that's where perhaps you can take, say, what's known as a gene drive. It's a particular type of genetic material that attaches to the mosquito DNA, and it will change the way in which that mosquito can cause viruses or parasites, in the case of malaria, from developing and being able to be transmitted from that mosquito. And so either you can block them from getting the mosquito from getting infected in the first place from the pathogen, the parasite, the virus, or it can help to prevent the mosquito from transmitting it on, or it may just cause a lethal gene, which means that if you introduce this genetic material into a mosquito, say in the immature stage, then when they are released in the environment and they mate with naturally occurring mosquitoes that don't have that gene, the gene is transferred to the other, to the wild type female, and it causes mortality either in the female or the next generation. So there's a lot of strategies around genetic modification. It's a little complex, but there's maybe 5 different strategies around modifying genetically a population of mosquitoes, releasing them, having them mix in the natural population of the mosquitoes in the area, and either causing mortality, death of the mosquito, or modifying the way in which they might be able to transmit the pathogen. So that's a conversation that's happening worldwide. In Mexico, there's every country's autonomous in their decision, on whether or not this is a viable strategy for them. In the U.S., we had some releases in Key West on some particular mosquito species that are representing GM strategies, and that came with some mixed feelings. But I'll stop there. Maybe Ildefonso, if there's time, might be able to provide some insight specifically around Mexico.
Yes, if I have some time, just a couple of minutes. What we saw in Tapachula, Mike, was mosquitoes sterilized by irradiation. It's a different approach, but they also sterilize the mosquito males. So you release these males in the field, they mate with wild females, and there's no babies. So that approach is safer, as is according to the WHO and other agencies. It's safer because it's not a pathogenic monster. So the method is very well seen to sterilize fruit flies to prevent mango and other fruits to be infested. So this is really good, and I see what's happening now in vector control, and that's why these flying mosquitoes will be very important. There were a report of 2 dead field technicians in Mexico. They were getting into the houses to check for mosquito breeding sites and also to spray indoor walls. They got COVID, and these guys died. So that means vector control indoors and also to apply repellents will change definitely, because you will not be in a house with and where you see an elderly person coughing, for instance. So I think we will, the release of flying mosquitoes on the neighborhoods, on the roofs, will be the following strategy. That's why I'm seeing, with all my love, these transgenic mosquitoes or sterilized mosquitoes with Wolbachia or whatever. So that's important. COVID is changing many, many things. And you see this example for indoor spraying in Mexico, and I think that it will be suspended, or at least a new strategy will be used. And the good thing is that we need to go to see the transgenic mosquitoes or the Wolbachia mosquitoes or all the mosquitoes as a good strategy, using drones, for instance, or any other aerial strategy, and to keep the health and the safeness of technicians. That's my commentary.
Thank you very much. I see we're past an hour already. So thank you to John and Nicole and Ildefonso and everyone else who joined. I know I really enjoyed this presentation, this conversation about all the wonderful work you're doing, specifically here in Mexico. So thank you very much for this.
You're welcome.
Thank you. Thank you, everybody. Thank you, Mike.
Thank you, everyone.
Bye.
And I'll just let everyone know real quick that we will be continuing lectures into the fall. So watch your emails. We'll be letting you know about new topics to come in the future. So thank you, everyone, and have a great day.
Great.
Thank
Thank
you.
you. Thank you, Mike.
Bye-bye.
Take care, everybody.